Hello, good morning. This is Ibukwan Folami, and today we'll be looking at how everything basically adds up together and how all this happens in the complex structure of the mitochondria in the process of the electron transport chain. So, what is cellular respiration? So, cellular respiration is the process in which ATP is produced through the metabolism of glucose. This is what we call cellular respiration. So many times some people confuse cellular respiration with breathing. So the fact that I'm breathing, taking in oxygen and breathing out CO2, well, is this really respiration? Respiration is actually how the oxygen I'm taking through my alveoli, how that oxygen combines with glucose to produce energy and water. And of course, it's good to memorize this equation, like I said, so that you can use it to understand what happens during cellular respiration. Of course, the CO2 produced is what we breathe out. So, how is energy generated from carbohydrates? There are about four important pathways, or I'll call them metabolic pathways. But they are basically biological reactions or biochemical reactions that happen. So energy is generated. We generate energy through glycolysis, and in glycolysis we produce pyruvate from glucose. We'll look at that in detail. We generate energy, through the Acetyl-CoA formation, the citric acid formation. This citric acid formation is also called the tricarboxylic acid cycle. It is also called the Krebs cycle. They mean the same thing. And of course, the electron transport chain. Now, we'll be going into the details of how energy is produced in these processes, through these metabolic processes, but keep in mind that you have actually done this course, most of you, so it should be a revision, kind of. So, we'll be looking at what happens in glycolysis. So in glycolysis, you have, glucose being converted to pyruvate. So glucose is broken down into two molecules of pyruvate. And what happens in the process, you have two ATPs being produced, and you have nicotinamide adenine dinucleotide, which is actually a high electron carrier. So we need to get familiar with these electron carriers as we are meeting them. NAD and NADH. We talked about the reduced form and the oxidized form. So NADH is the reduced form, and it's produced also when glucose is converted to pyruvic acid in glycolysis. So, through glycolysis, we convert glucose to pyruvic acid. We produce 2 molecules of ATP and 2 molecules of reduced NAD. This is important because many times we use this pathway to produce short supply of energy So if you need energy, maybe a quick energy, you can put it like that, the body can produce it through glycolysis. But it's not really sustainable in the sense that if you need a lot of energy, this metabolic process is insufficient to produce energy in a long term or in large supply. So the second step is the formation of a metabolic intermediate that we call acetyl-CoA. So after glycolysis, the pyruvic acid produced is used to form acetyl-CoA, and acetyl-CoA produces carbon dioxide, but it also produces 2 molecules of NADH. It's important to note that yes, all this NADH produced, so the high electron carriers, are important because somehow the body uses them. We'll see later how the body uses them, all these electron carriers, to actually produce ATP in the electron transport chain. But you need to understand that yes, all the high electron carriers like NADH produced will still be used in the electron transport chain to produce ATP. But we'll get there in a short while. Now, the third pathway is what we call the TCA cycle or citric acid cycle. Now, in this cycle, the acetyl-CoA produced during the intermediates, remember we looked at it earlier on, so it's an intermediate compound that is produced, goes through a series of metabolic reactions, and in this series of metabolic reactions, the important part to consider is that NADH is produced and another electron carrier, we are meeting this for the first time, is also produced at different points. So we see here between the point in which isocitrate is converted to alpha ketoglutarate, NADH is produced. The same way, between the point in which alpha ketoglutarate is converted to succinyl-CoA, NADH is produced. These are electron carriers, and we meet here between the point in which succinate and fumarate is produced, FADH is produced. FADH stands for flavin, adenine, dinucleotide. And like NADH, it's also a high electron carrier. We'll meet it during the electron transport chain. Like NADH, it works in it exists in 2 forms: the reduced form and the oxidized form. So just like NADH, when the reduced form loses its hydrogen, it becomes oxidized to FAD. And the same way, when the oxidized form gains electrons from hydride ion, it becomes reduced to FADH. Okay, so what is important about the citric acid cycle? It is important to know that in the citric acid cycle, the body generates energy. It happens where the citric acid cycle happens in the matrix of the mitochondrion, and through the TCA cycle or the citric acid cycle, the body generates these high electron carriers, NADH and FADH, and the high electron carriers are precisely what is used to generate ATP later on in the electron transport chain. So far, let's begin to account for all the energy metabolites that we have produced. So in the citric acid cycle, you generate 3 NADH, 1 FADH, and 1 ATP. This is very important. Remember, we also did the same for glycolysis. When glucose was converted to pyruvate, we generated 2 moles of ATP and 2 moles of NADH. It's important that we account for the ATP and also the electron carriers, because like I said, these electron carriers is what is going to be used to generate more energy in the form of ATP in the electron transport chain. So a bit on where things are happening. So the question is, where are all these reactions going on? So glycolysis occur in the cytoplasm. So think about it. You have here your mitochondrion. Where is your mitochondrion? Your mitochondrion obviously is in the cell, right? So in the cell, it's always good to remember that the cell is a phospholipid bilayer, and 2-dimension structure is usually represented with a double membrane. Inside that cell, obviously, we call the interior of the cell the cytosol or cytoplasm. So I'm putting this in context. So glycolysis occur inside the cytosol. It's good to remember that. But of course, the TCA cycle or the Krebs cycle occur in the inner matrix of the mitochondrion. So it's also good to see the context of where these reactions are occurring. We continue. So this also depicts exactly where things are happening. It's not as descriptive as the previous slide, but it explains exactly the same concept. So we are looking at glucose now, but of course you also see the trend for fats, but we are considering sugars, polysaccharides, glucose, glycolysis in the cytosol, but of course pyruvate is transported from the cytosol into the mitochondrial matrix, and acetyl-CoA is produced in the mitochondrion, and from there all the reactions which we considered earlier on are produced also in the matrix of the mitochondrion. Okay, so at this point we look at the electron transport chain, and yes, this is basically a diagram showing the key enzymes of the electron transport chain. So this diagram where is it? What's the context? Because sometimes we see this diagram and it's sometimes difficult to interpret. But we realize that we are still very much in the matrix, or we are still very much in the mitochondrion. So don't forget, in the mitochondrion we have a double membrane, and this is the outer membrane of the mitochondrion. But we also have an inner membrane that folds into the cristae, and the inner membrane is what makes up the folding, and the inner membrane inside it is what we call the matrix. So the point here is, where exactly is all this structure? So where exactly does it represent in the mitochondrion? This is the inner membrane of the mitochondrion, and this is where electron transport chain occur. This is very important, that what you have here is the inner membrane of the mitochondrion, and where does it translate to in this two-dimensional structure? It is precisely this double membrane. So we can say that this double membrane is zoomed to produce this. So when you zoom inside the double membrane, the inner double membrane, the phospholipid bilayer in the inner membrane, this is what you have. In the electron transport chain, the electrons that are produced are transferred across the inner membrane proteins. And during the transfer of these electrons, a current is passed along the membrane. And you understand that we talk about current flow when electrons are moving along a conductor. So we can imagine a similar thing happening along the membrane of the matrix. So these proteins in the inner membrane of the mitochondrion are called complex 1, complex 2, complex 3, complex 4. So they have their biochemical names, which we should be aware, we should know them. So complex 1 has its name, complex 2 has its name, and complex 3, they have other names that they are called. But let's keep it simple, although I'll require you to know those names, but let's keep it simple. So in complex 1, you recall that NADH in complex 1 is oxidized to NAD+, and in the process you have the proton ion or the hydrogen proton, and you have 2 electrons that is released. So these 2 electrons are precisely what is passed to complex 3, and of course this keeps on happening. So you need NADH in that sense to generate these 2 electrons. Hence, that's why we call NADH, FADH high electron carriers. As NADH is generating the 2 electrons, the proton produced is pumped by this protein, complex 1, into the intermembrane space. So this is quite important, and in a similar way for FADH2, another high electron carrier, in a similar way FADH2 is oxidized to FAD. The electrons produced are transferred to complex 3. Meanwhile, the proton produced is passed into the intermembrane space. So the question is, why are we generating these protons, and why are these protons being pumped by these proteins into the intermembrane space? It is because at the end of the day, we want to carry out what we call oxidative phosphorylation. So during oxidative phosphorylation, what happens? During oxidative phosphorylation, as a result of the proton gradient, there is an electrochemical gradient that is formed across the membrane. Why? Because in the intermembrane space, there are so many protons compared to the inner matrix of the mitochondrion. As a result, there is an electrochemical gradient created, and this gradient, the electrochemical gradient, is used as a kind of power, and this gradient powers this enzyme called ATP synthase. So the electrochemical gradient is used to power ATP synthase, and in the process of powering this, you need to see the video. You need to really see the video to understand it. In the process of powering the ATP synthase, it generates ATP from ADP, and actually this is how ATP is generated in the animal cell.